Welcome back, everyone. We finished discussing some prokaryotic mechanisms of gene regulation, which may somewhat carry over into eukaryotic organisms. But let's start talking about us specifically, our bodies as humans. So we are multicellular organisms. We have lots of different cells, and those different cells are different from each other. They have been differentiated or specialized for their different cell types. So there are various types of stem cells which are at various levels of not yet differentiated, which can then give rise to different types of cells. So muscle cells, sperm and egg, immune cells, epithelial, nervous cells, blood cells, bone cells, fat cells. All of these are different kinds of cells that your body can make, and they're different from each other. They've been specialized to make the proteins that they need to have the morphology that they need to function, and are not producing the proteins that other cell types may be. So they have different morphology, they have different protein products. And the reason why they look like that is because they have differential gene expression. They're creating different proteins based on differential gene expression. They have different shapes and structures because of different gene expression, because they're able to turn on genes that they need and turn off the genes that they don't need. There's really 5 different levels of gene control where you can control what proteins are being made. The first one is at the level of the genome. So in very rare cases, genes can be amplified or deleted to make more or less of a specific protein, or DNA can be rearranged. Those are very rare because those are obviously going to be very permanent and passed on to future generations. That is probably not always what the cell wants to do. Some other ways we can control genomic level of gene expression: DNA methylation, so adding a methyl group to a nucleotide, which can affect its ability to be expressed. Condensing and decondensing chromatin, so wrapping it up tight where transcription machinery cannot access the DNA, or letting it be a little bit more loose where that transcription machinery then can access the DNA and transcribe that gene. Histone modifications, things like methylating or acetylating the histones, which then will relate to its ability to be condensed or decondensed and its ability to be expressed. And non-coding RNAs like exist, which is a way that we wrap up our X chromosomes into bar bodies. So then you have genes available for expression at that level. Then we can also control at the level of transcription. This would be by having different transcription factors, having different genes being transcribed or not. Then you can control at the level of RNA processing and nuclear export, splicing differentially different RNA products, other different types of processing your RNA, and then transferring that mRNA out of the nucleus into the cytoplasm where it has to get to be translated. Then next we have the level of translation, once that mRNA is in the cytosol. One is by degrading mRNA. So if the mRNA is going to be degraded quickly, you're going to have fewer protein products. And if the mRNA is staying around, existing longer, you'll have more protein products. Other things we have are targeting newly forming polypeptides to the endoplasmic reticulum, controlling translation by initiation factors and translational repressors, microRNAs, which can affect the ability to make the protein. Then once a polypeptide is made, there's post-translational control. Things like protein folding and assembly, which have to occur for the protein to be functional. Maybe cleaving your polypeptides to either activate it, like we talked about with things like insulin can be activated by polypeptide cleavage, or cleaving a polypeptide so that it is no longer functional. Modifying a protein, we've talked about that a lot this semester. Things like phosphorylating, dephosphorylating, ubiquitinating, acetylating, glycosylating, different things that can affect that protein's function. Importing it into organelles for it to be functional, and then degrading the protein. So these are all different ways that your cell is able to control what proteins are right there currently in use in that cell, what kind of morphology, what shape it has, what structures it has, based on all of these. We're going to spend the rest of this chapter discussing some of these key differences and how they allow proteins to be expressed or not in your cells. So let's talk about cloning first. In 1964, there were nuclei from tadpole cells. So these were already differentiated tadpole cells. And the nuclei from those tadpole were put into unfertilized eggs. So instead of taking an egg and a sperm to create a diploid egg, they just took a diploid nuclei from a tadpole cell and put it into that egg. The egg had its original nucleus taken out of it. So now you had an egg cell that was diploid. With the theory being that then would that egg grow into a tadpole like any other diploid cell from fertilization would grow. The success rate was very low, but some of those eggs did develop into swimming tadpoles. They still had to go through the process of embryonic development like any other egg would. But those tadpoles, that is nuclear transplantation, when you transplant the nucleus of your tadpole cell into the nucleus of an unfertilized egg. So that meant that these tadpoles that grew were clones of that original tadpole. So because it had the exact same DNA from that tadpole, they were all clones of each other. In 1997, a sheep was cloned, Dolly the sheep. This is the first mammal that was cloned, and this used nuclear transfer, or they took the nucleus of an egg and took it out of that egg cell. They took a nucleus from an adult sheep cell and put it into that egg cell. So now you had a diploid egg. That egg was transplanted into a surrogate mother where the egg developed into, through embryonic development into a little baby sheep that was ready to be born into this little sheep Dolly. So Dolly, or the mother, donated her DNA into this egg. So the egg was then transplanted into a surrogate who gave birth to Dolly the sheep, who was a clone of the original sheep who donated the cells. So this showed that the nucleus of a differentiated cell is still able to direct the development of the entire new organism. So even though that cell was differentiated, it had not lost any of its DNA. All of that DNA was still there in that differentiated cell. And that holds true for all of the cells of your body. Your cells, every cell of your body still, with the nucleus, every nucleus of your cell still has all of that DNA in it. That's able to be turned on and turned off. So your nucleus is totipotent. That means it has all of the genes that are needed to create a new organism of the same type from which nucleus was taken. Reproductive cloning of humans is not something that we currently do. It's really difficult. So, for example, it took hundreds of attempts to clone Dolly the sheep. There's also health problems, a lot of health issues to deal with. A lot of cloned animals die prematurely, have a lot of health problems. We still have a lot to learn about things like telomeres and aging and healthy development of cells. And there's also a lot of ethical problems. People have ethically they have a hard time sometimes thinking about cloning. Or I think also a lot of when you don't understand science as well, and we live in a culture where that is not understood well, that can also cause problems with understanding how things are how science is working. One possible application of the cloning technique is nuclear transfer technique. And this is to produce stem cells that are genetically matched to a patient. This will be used for therapeutic cloning, so not to create a new human, but to create new cells that match your patient so that they can replace their damaged cells. Currently, we can do things like transplants that require a patient to be on immunosuppressant drugs for the rest of their life. But therapeutic cloning, because those cells are genetically matched to the patient, would not elicit the same kind of immune responses. You can take a stem cell, and your stem cells can replenish themselves through continued division. Your stem cells are not differentiated, or they may have only begun to be differentiated. So they're able to produce a variety of different kinds of cells that can then differentiate in response to the signals and then create, so here in this picture, they took a fibroblast, de-differentiated it, used an oocyte to create embryonic stem cells, which created a muscle cell. So this patient is able to have a muscle cell replacement. So stem cells from mature organisms are multipotent. That means they can form several cell types. So you may have a multipotent cell that can create different types of bone cells or different kinds of blood cells. So you're able to make new B cells and platelets, but that multipotent cell isn't going to create other parts of cells. Embryonic stem cells, on the other hand, are pluripotent. That means that they can come and create all the types of cells that give rise to a human organism. That, I guess, is probably pretty obviously a requirement of being an embryonic cell to be able to differentiate into all of the cells of a body to create a growing person. They just can't give rise to the cells that form the placenta and other support membranes needed during gestation. Embryonic stem cells come from a group of cells called the inner cell mass. They are found at one end of the early embryo, one end of the blastocyst. And the ability to isolate and grow stem cells in the lab raises the possibility of producing new healthy cells to replace damaged cells in patients with diseases. But currently, this is something that is ethically, there's a lot of ethical concerns around it. Harvesting embryonic stem cells, and a lot of debate about when life begins. That really makes using embryonic stem cells not a reality for almost everyone in the United States. But there is something called iPS cells. This is a way that makes cells that act like embryonic stem cells, but they don't come from an embryo. So you don't have those associated ethical issues of embryonic stem cells. So this lab took differentiated cells from mice, and they were able to revert it to a pluripotent state. So that's where iPS comes from, is they're induced pluripotent stem cells. And these came from the adult, the donor, but because they have been de-differentiated, they have a lot of properties similar to embryonic stem cells. However, there's a lot of issues that still need to be overcome with that before they are used as therapies. But it is something interesting in your textbook. If you want to read about, you can get more information there. Okay, then our next topic we're going to talk about in terms of gene regulation is how lymphocytes, or white blood cells, are able to rearrange their genes. This is especially important in terms of producing antibodies, that you are exposed to millions of different types of potential pathogens. And so by being able to rearrange those genes, your body is able to create millions of different types of antibodies. The first way we can create variety within our white blood cells producing antibodies are that antibodies are composed of heavy chains and light chains, these two different types of polypeptide subunits. So we can create different types of heavy chains, different types of light chains, and put them in different combinations. So already by having two different kinds of subunits, you have a little bit of variety within your antibodies. However, we're able to produce millions of different types of antibodies, but we don't have millions of different antibody producing genes. So there's a lot more intensity going on in terms of how we're creating our antibodies. So lymphocytes are going to use a small number of genes. We have about 200 V segments, about 200 D segments, and about 20 D segments and about 6 J segments. And those are going to be combined to create millions of different antibody genes. There's also a C region, although that constant region is going to be constant, like it is not going to be varied. whereas the V, D, and J segments can be put together in unique combinations. They are the variable regions of your antibody. So out of these 200 V segments, 20 kinds of D segments, 6 kinds of J segments, we can rearrange these during lymphocyte development, and every white blood cell is going to rearrange these segments in unique different kinds of ways to create about 24,000 different types of heavy chain variable regions. So here this one has come up with this variable region. It goes through DNA excision, randomly removing several different D and J segments. Then another set of random excision, removing several V and D segments. Then transcription, bringing together your pre-mRNA. And then RNA splicing, removing more segments to create a unique final mRNA. Light chains are also able to be variable. They don't use D segments, so there's about 1,000 or thousands of different types of combinations you can get for your light chains. And then as those random heavy chains and the random light chains are put together, you'll end up with millions of different types of antibody combinations being produced. All right, we've got some more to talk about how we are creating different proteins in our cells. So make sure you come back for our discussion to continue, and I'll see you later. Bye.